Okay, so in the previous section, we saw that proteins are polymers of amino acids. And in the previous section, we looked at those building blocks, the amino acids, and saw what they themselves were made up of. Now we're going to see how those amino acids are linked together. So when we start linking and connecting those amino acids, right, we're going to link them together. Amino acids are connected by what are known as peptide bonds. Okay, and a peptide bond is nothing more. It's the same as an amide bond, which we've seen in a couple chapters ago, right? And if you recall, in order to form an amide, right, an amide bond forms when a carboxylic acid and an amine react together. Well, guess what? What functional groups does an amino acid contain? Well, it contains an amino group and it contains an acid group, right? So an amino acid actually has all the right ingredients to make these amide bonds, which in a protein we just call a peptide, right? Peptide and amide mean the exact same thing. So some terminology. When we link two amino acids together, we call it a dipeptide. If we link three amino acids together, we call it a tripeptide. If we link four amino acids together, we call it a tetrapeptide. And you can kind of continue this. If we had 5 amino acids, we would call it a pentapeptide. And I'm just going to say ... All right, till you string enough of these together that we just generally call it a protein. So how does the peptide bond actually form? Well, we just said you need to combine the carboxylic acid with an amine. Well, let's take a look at how that can form. And we'll just choose 2 simple amino acids here. We have glycine and we have alanine. All right, so we draw both of these. And generally when you draw your amino acids, typically, okay, the amino group is on the left of the amino acid, and then the acid group is on the right. Right, that's a convention that we use. So there's our glycine here on the left, the first amino acid, and then we have our alanine right next to it. So when we combine these two, right, we can see right away that we already have the carboxylic acid group from 1 here, the glycine, right next to the amino group of the neighboring amino acid. And what's going to happen, right, is we are going to end up losing or getting rid of or generating water as one of the products, as 2 of those H's from the amino group combined with the O from the neighboring amino acid to form our water, which is this. And then all we do is we're going to link together now this carbon, nitrogen. So we end up forming our brand new peptide bond, which is this bond here, right? And we still have our glycine on the left side, this part of it. And then we have our alanine component on the right side. So what you will notice is when we start linking these amino acids together, one end of the growing peptide now is an end that has a free nitrogen. It's not linked, or it's not part of an amide bond, and we call this the N terminus. And then on the other end, we have our free carboxylic acid or carboxylate. We call that the C, the carbon terminus. Right, and every single peptide is going to have an N terminus where the peptide starts, and then a C terminus where the peptide ends. Right, so the N terminus starts the peptide, and then the C terminus terminates or ends the peptide. This may not seem like a big deal, but it actually is a huge deal when it comes to looking at how the amino acids and in which order the amino acids are linked together. All right, so we form a dipeptide here because we link two amino acids together. Okay, so this then we would call a dipeptide. Not because there are two peptide bonds, but because there are two amino acids, right? And that's a common misconception that students have at the beginning. Dipeptide means not two peptide bonds, but two amino acids. All right, so let's see if we can practice that here. We have serine and we have threonine, and we want to draw the dipeptide between them. So let's expand this out a little bit. So we have our NH on one side. From the serine, our CH. There's our R group, and then we have our our carboxylate end, and we're going to combine that with our NH. Three plus from the other side, our threonine. And then there's our R group for threonine. There's a CH with an OH. And then there's a CH3 up here. So I'm just going to highlight the R group in each of these. All right, so we're going to do the same thing like we did in the example. We're going to combine the O here from the serine with those two H's from the threonine, so we end up losing water. Two H's and an O. And then what we're going to draw again, draw everything that does not change. So we have our NH3 here from the serine, our CH, and then our R group, CH2OH. Our C double bond O, and now we're going to connect this C double bond O to that nitrogen from the threonine, and then to that nitrogen we have our H underneath, we have our C, that's our alpha carbon, and our CH3 on top, and then we have our C terminus on that side. All right, so we can highlight our amide bond. Here it is. And that's the dipeptide that we would make in this case. All right, so we still have our serine on this side and our threonine on the other side. And then we still have our R groups highlighted here. All right, and just to complete this, our alpha carbon, we can highlight in yellow, right? The alpha carbon is always connected to that R group. All right, so in green here, this is our R group. And then in yellow, we have our alpha carbon. Right, so I'm going through this because this is a really good way to approach your peptides, is being able to identify where that alpha carbon is, because we know what the R group is going to be, and then we can identify the amino acid as a result of that. All right, so that's a pretty simple peptide. It's only made up of two amino acids, so it's a dipeptide. And they're connected serine first and threonine second, right? So the other thing here is there's our N terminus, and then there's our C terminus. right? So there is an order in which we connect the amino acids, and that's key, as we'll see in a second. When it comes to naming the peptides, we're not going to get too deep into this. We're not going to name really long peptides systematically. We're just going to limit ourselves. So the way that we name our peptides is we're going to take the names of the amino acids at the beginning, and we're going to change the ending to YL. And then the C terminus, the C terminal amino acid, we're just going to keep the name as it is. All right, so if we have our serine and threonine that we drew before, all right, we have serine connected to threonine. The way that we would name this, we would drop the ending, so this becomes seryl threonine as the name. All right, and if we had something a little bit here, we have our alanine, glycine, and serine. All right, so let's take a look at this one. We have alanine, glycine, and then serine. Okay, so we're going to drop the ending for the first two. We'll replace it with YL, so we have A, alanyl, and then glycyl, and then it ends with serine. All right, so we change the ending for the first amino acids, and we keep the full name for the very last, the C terminal amino acid. All right, let's take a look now at talk a little bit more about the amino acids. We mentioned that there are 20 amino acids that are used to build proteins in the body. Out of those 20, 11 can be made by the body, which means the body can use whatever it already has to make those amino acids. The other 9, right, the body cannot make, and you must get them from your diet. And we call those 9 amino acids essential amino acids. So essential amino acids must be obtained from the diet, right? And you take a look here at these essential amino acids. So when you eat protein-rich foods, right, you want to make sure that you have complete proteins, meaning that they are going to contain all of those essential amino acids that your body actually needs. So you'll notice that typically meat products and fish products contain essential amino acids, whereas proteins that you get from plants are incomplete, right? Meaning that they're going to be missing or they're deficient in some of those amino acids. All right, so now we're going to jump into, and we'll continue this discussion in the next section. We're going to take a look at what's known as the hierarchy of protein structure. Hierarchy, spell that correctly. Okay, and what that means is that there's some sort of a system to how we're going to look at the structure of your proteins. So we're going to be taking a look at primary structure that determines secondary structure, that determines the tertiary structure, and then we may or may not have a quaternary structure, right? And we abbreviate these: primary 1 with a degree sign, secondary, just as we did with our alcohols and amines, and we have our quaternary, which is abbreviated like that. All right, so the first one we're going to take a look at is the primary structure of proteins. And we're going to say that the primary structure of protein of a particular protein is just the sequence of the amino acids. All right, so the primary structure is simply equal to the sequence or the order of the amino acids, and this is the key part now, from the N-terminus to the C-terminus. All right, so if you just list the amino acids in that order, all right, that is what the primary structure actually is. All right, so if we take a look, let's look at this one. Actually, we'll focus in on the one underneath. All right, so if we take a look at a fairly simple, this would be a tripeptide underneath here, made up of glutamate, histidine, and proline. All right, if we were to write the primary structure for this, right, all that the primary structure is to list the amino acids from the N-terminus to the C-terminus. All right, so we're going to go in that order, which means you have to identify these amino acids. So how are you going to identify the amino acids? Well, you want to first of all locate the alpha carbon, right? Because remember, it's the R group that determines what that amino acid actually is. So the amino acid is connected, the alpha carbon is connected to both the amino and the acid group. So we can maybe circle our alpha carbons here in red. Right, and you'll notice that all of them are connected on one side to a nitrogen, on the other side to a C double bond O. So that means then we can look and see what else is attached to that alpha carbon. We can highlight it in yellow, and you'll see proline is an unusual case where it actually folds back upon itself. So we can use then our ability to identify the alpha carbon to say what amino acid we're dealing with. So in yellow here we have the R group. We can look that up on our table, and we can identify this as being glutamate, histidine, and proline. So our primary structure then would simply be to list the amino acids in that order from the N-terminus to the C-terminus. All right, so it'll be glutamate, we have histidine, and then we have proline in that order. And it's as simple as that. When you talk about the primary structure, it's just listing the amino acids from the N-terminus to the C-terminus. Okay, so let's take a look at an example, another one here. So we're given a structure, and just by looking at this, right, you may think, oh wow, this looks so confusing. So the first thing that you want to be able to do is to orient yourself, and in most cases, all right, you'll see that the peptide is going to be given to you in the order from the amino end to the C-terminus end. This is a typo here. There should be NH3. Okay, so you want to start systematically here and work from left to right. So we know that this amino group has to be connected to the alpha carbon. So our first alpha carbon is going to be here. All right, that alpha carbon is connected to a C double bond O, the acid part of it, and then that is going to be connected by our amide bond. And we're going to highlight our amide bond in green, so C double bond O N. And if you look further down, you can see your other amide bond over here. All right, and then in between those amide bonds, well, what's going to be in between them? Your other alpha carbon. And then on the tail end of this, if we go beyond that second amide bond, we'll have our third alpha carbon right here. All right, so the red circle is the alpha carbon, which means now looking at our R groups, what's attached to that alpha carbon, we can identify what the amino acid is. All right, we can highlight those in yellow. All right, so our first amino acid here, this is phenylalanine, PHE. This R group is for cysteine, CYS, and then this Our group here, which is just a methyl group, is for alanine, ALA. All right, so you have, when you identify the amino acids, you have to go through it systematically, and what can help you do that is to locate the alpha carbon, right? And what will point you in that direction is to find your amide bonds, which you already know what an amide bond looks like, right? We did that in the earlier chapters. All right, so we have our, now we can answer the questions. What's the amino acid at the N terminus? Well, that is going to be your phenylalanine. The amino acid at the C terminus is going to be your alanine. And then if we wanted to, right, we can write the primary structure for this, right, from N to the C terminus is going to be phenylalanine, PHE, connected to cysteine, connected to alanine. All right, and that's all that the primary structure is, just a sequence of the amino acids in that order. And that's essential when it comes to primary structure. Because if you change that order, you change what that peptide is, right? The order makes a huge difference. And what I mean by that is phenylalanine, cysteine, alanine is not the same as, so that's not equal to phenylalanine, alanine, cysteine, right? They're different. And that is not equal to alanine, phenylalanine. So they're all made up of the same 3 amino acids. The order is different, right? And that means that the peptide is different as well. Okay, so the primary structure is essential when it comes to determining all the other levels of protein structure that we'll see subsequently.